This is Chapter 18, Cardiovascular System on the blood vessels. So last chapter, in Chapter 17, we looked at the heart. So this chapter, we're going to look at the blood vessels that are going to carry the blood all throughout the body that the heart pumps. So the blood vessels are our body's delivery system that's going to begin and end at the heart. So it's going to make a complete circuit all the way through the body. It's also going to work in tandem with the lymphatic system to help circulate these fluids. And we'll talk about the lymphatic system in the next chapter, chapter 19. So there's three categories of blood vessels in the body. Right? So the arteries are going to carry the blood away from the heart. Okay? Generally, this will be oxygenated blood, denoted with red. The only exception would be in pulmonary circulation, which we'll talk about later on in this chapter, and also in circulation of the fetus, since their lungs are not active yet. Capillaries are the only vessels that have direct contact with the tissue cells, and they're going to help directly serve cellular needs by delivering oxygen and nutrients and removing any waste products like carbon dioxide. The veins are the vessels that carry the blood back toward the heart. And generally, they will be deoxygenated blood, denoted as blue. Again, the exception would be the pulmonary system and the fetal circulation. So the pulmonary circulation is a little bit backwards or opposite from systemic circulation that we'll talk about later on in this chapter. So looking at the structure of the blood vessel wall, so all blood vessels will contain a lumen, which is just the opening in the middle of the blood vessel where the blood is going to flow through. So the walls of blood vessels, with the exception of the capillaries, will consist of three layers or tunics. So we have the Tunica Intima, the innermost layer, Tunica Media, which is the middle muscular layer, and the Tunica Externa, which is the external connective tissue layer. With capillaries, since they're so small and thin, they only contain an endothelial lining. So we want that thin blood vessel wall so nutrients and things can pass easily between the tissues. So looking at the Tunica Intima first, the innermost layer. So it's called the Tunica Intima because it's in intimate contact with the blood. Okay? So it's going to consist of an endothelium, so an inner lining tissue. So it's going to line the lumen, the inner chamber of all blood vessels. It's also going to be continuous with that endocardium, so that inner lining of the heart. And it's composed of a slick surface, so we can reduce friction and resistance to any blood flow. The subendothelial layer is just a connective tissue membrane that's going to help stabilize that endothelial layer. And this is generally only found in vessels that are larger than 1 millimeter or so, then they need a little extra support. The Tunica Media, we said, was the middle layer, composed mostly of smooth muscle and some elastic fibers. So that's going to allow the blood vessels to stretch and recoil as blood is pumped through. So these are innervated with sympathetic nerve fibers that are going to control vasoconstriction and vasodilation. So remember, the sympathetic system is your fight or flight, so it helps to increase blood pressure. So an increase in sympathetic impulses would increase vasoconstriction, which would decrease the diameter or the size of the lumen opening, which would increase your blood pressure. Or vice versa, if we decrease our sympathetic impulse output, the vessels would dilate to increase the diameter and reduce blood pressure. So the Tunica Media has the greatest influence on blood flow and blood pressure because it's going to control the resistance to that blood flow. The Tunica Externa is that external outermost layer. Sometimes it's referred to as the Tunica Adventitia. Adventitia is just a surface lining structure. So it's going to be composed primarily of collagen fibers to help protect and reinforce the vessel wall and also anchor it to the surrounding structures. So as you can see, it's infiltrated with nerve fibers, smaller blood vessels, lymphatic vessels. Okay. So these small blood vessels within the blood vessel are called the vasa vasorum. So these are going to nourish the larger blood vessels themselves, that outer layer, tunica externa tissue. Okay, so this is just kind of a comparison summary of the different types of blood vessels. So we'll talk about the different types of arteries and veins shortly, but just looking at relative makeup of the blood vessel tissue in terms of smooth muscle versus elastic tissue. So arteries, we see, have much more smooth muscle than the veins, and they also have much more elastic tissue. Because they deal with higher pressures, they need to accommodate that stretch of the vessel wall. Okay, so looking at the arteries first, we can divide these into three groups based on their size and function. Okay, so we have elastic arteries, muscular arteries, and arterioles. So looking at the elastic arteries first, so these are the ones closest to the heart. So they have very thick walls with large, low resistance lumen. So coming straight off of the aorta, they're going to have to deal with high blood pressure. Right? So it's coming fresh out of the heart, freshly pumped. So the blood pressure is going to be highest in these elastic arteries. So they have large amounts of elastin or elastic fibers to allow them to expand and recoil as that blood is ejected from the heart. Okay. So their large open lumens are also going to reduce resistance to flow, so the blood flows very freely. They're just kind of open elastic pipes or tubes. So this helps to allow for continuous blood flow even between heartbeats. And again, they contain a lot of smooth muscle, but they're not really important for vasoconstriction because they have such large lumens. They're not able to completely constrict enough to slow down the blood flow. So these elastic arteries, as they go throughout the circulation, are going to branch off and diverge and get smaller and smaller. So they're going to give rise to the muscular arteries. Right? So these are sometimes called the distributing arteries because they're what's going to actually deliver blood to the major organs. Okay? So most of the named arteries that you'll have to learn for lab will be these type of muscular arteries. So being that they are muscular arteries, they're going to have the highest amount of that smooth muscle tissue and a thicker tunica media, but with less elastic tissue. So because they do have that thick muscular layer, they are active in vasoconstriction. So they're able to constrict and decrease the size of their lumen or opening to slow down blood flow. So then the third type are the arterioles. So these are the smallest of the arteries. So while the larger arterioles contain all three tunics, so the Tunica Intima, the Tunica Media, the Tunica Externa, the smaller arterioles, as we get closer to the capillary beds and the tissues, are mostly just a single layer of that smooth muscle surrounding some endothelial lining cells. So these arterioles are what's going to help control the blood flow into those capillary beds either by vasodilation or vasoconstriction. So if we want to increase the amount of blood flowing into a tissue, we can vasodilate and increase the diameter of our blood vessels. So you can think of it as a highway, right? So if we open up another lane of traffic on the highway, right, so we dilate, right, so traffic will move more smoothly and more quickly, as opposed to with vasoconstriction, we're closing some lanes in the highway, so there's going to be a little bit of slowdown in the traffic. Right? So these are sometimes also called resistance arteries because they're able to change their diameter so dramatically to change the resistance to the blood flow. So the third type of blood vessel we'll look at are the capillaries. So these are microscopic blood vessels, like we said, that have direct contact with the tissues. Right, so some of these can be so tiny that red blood cells can only pass through single file one at a time. So their functions are gas exchange, delivery of nutrients, removal of waste, hormones, etc., between the blood and the interstitial fluid. So remember, interstitial fluid is just the fluid between the cells and the tissue spaces. So with capillaries, the walls are just a thin tunica intima. And sometimes even in the smallest vessels, only one cell can form the entire circumference or diameter of the blood vessel. So they're also going to contain some specialized stem cells called pericytes that are just going to help to stabilize the capillary wall and control permeability, and also play a role in any vessel repair. So we kind of have these cells on the outside to help support and stabilize this internal intima layer. So generally, capillaries will supply every cell and tissue in the body, except for a few select locations like cartilage, epithelia, the cornea, and the lens of the eyes, which are all avascular tissues. So capillary cells, the endothelial cells, are joined by what's called a tight junction. So we've talked about these before in Anatomy 1. Right? So tight junctions are where the cells are very tightly kind of stitched together, right, to prevent any excess fluid loss or seepage through those tissue cells. So the gaps in between these endothelial cells are called intracellular clefts. Right? So any small solutes and fluids will be able to kind of squeeze through these endothelial cells at these intercellular clefts. So there's three types of capillaries we'll look at. So the first ones are continuous capillaries. So these are the most abundant type in your skin, muscles, lungs, and the central nervous system. In the brain, they are a unique type of continuous capillary. So it's going to form that blood-brain barrier. So the endothelial cells will be very tightly enclosed, and they have little to no intercellular clefts. Second type of capillaries are fenestrated capillaries. So fenestra means window in Latin. So these capillaries have lots of fenestrations or little pores or windows in their endothelial cells to increase their permeability. So these are found in areas where we have a lot of active filtration, where the blood needs to pass through the capillaries relatively quickly and be filtered. So places like the kidneys, the intestines, and endocrine organs. The third type are the sinusoid capillaries. So these have very few tight junctions. They have some larger fenestrations, larger intercellular clefts, and generally incomplete basement membranes, and larger lumens. So these are considered the leaky capillaries. So think sinusoid, your sinuses. Right? So when you get a cold, your sinuses are leaky. So these are leaky capillaries that are only found in select areas of the body like the liver, bone marrow, spleen, and the adrenal medulla. So because they're so permeable, the blood flow is kind of sluggish. So this is to allow adequate time for those larger molecules in blood cells to pass between the capillaries and the tissues. Some of these will also contain macrophages to capture and destroy any foreign invaders. So since they are more open blood vessels, they're more likely for pathogens and things to make their way inside. Right? So we have kind of a protective barrier against those with the macrophages. So capillary beds are interwoven networks of these tiny capillary vessels between an arteriole and a venule. Right? So sometimes this is referred to as the microcirculation. So the flow of blood through the capillary beds in the tissues. So there's two main types of vessels in capillary beds. You have the vascular shunt vessels, which is essentially just kind of this straight across, straight through from the arteriole to the postcapillary venule. Right? And then the true capillaries that are involved in gas exchange are going to branch off and form kind of this interweaving network of the capillary bed. So looking at the vascular shunt, so it's going to go from the met arteriole through the thoroughfare channel. Right? So it's kind of a continuous straight point A to point B. So we kind of bypass the rest of the capillaries in these tissues. So the terminal arteriole, so terminal being like, the end, right? So the very last stop for the arterial pathways, the terminal arteriole, is going to feed into this met arteriole that's continuous with the thoroughfare channel and then drains the bed through the postcapillary venule. Okay. So again, red denotes oxygenated blood, blue denotes deoxygenated blood. So you can see as we pass through the capillary bed in the tissues, we're transitioning from red to purple to blue. So at this point, the tissues have taken up the oxygen from the blood and replaced it with carbon dioxide to be exhaled in the lungs. So the true capillaries are the actual exchange vessels. So these are the true capillaries that are going to branch off and form that network of the capillary bed. So they'll start to diverge off of that met arteriole and then return to the thoroughfare channel. So they're going to contain what are called precapillary sphincters to help regulate blood flow into these capillaries. So if the tissue doesn't require blood at this time, or we need to redirect blood to, say, the heart or the brain instead, we can close these sphincters so the blood is directed straight through that thoroughfare channel and the vascular shunt. Right? So the veins, the venous system, is going to carry blood back toward the heart. So veins are going to, formation of veins begins when the capillary beds unite and form those postcapillary venules. And then from there, they're going to merge into larger and larger veins. So whereas the arterial system branches off and diverges into smaller and smaller vessels, the venous system is going to merge into larger and larger vessels as we get closer to the heart. Okay. So the venules are the capillaries united to form those postcapillary venules. So these are just, think of the really small veins. Okay. So arterioles are the really small arteries. Venules are the really small veins. Okay. So they're going to consist of primarily just an endothelial lining and a few of those pericytes for support. So these venules, being very small, are also very porous to allow fluids and white blood cells to seep into surrounding tissues. So as we get to the larger venules, they will have one or two layers of smooth muscle cells, but they're not really going to play much of a role with vasoconstriction. So as the venules unite and converge into larger and larger vessels, we get to the veins. So the veins have all the same tunics as arteries, but their walls are much thinner. They have larger lumens generally, and they're more kind of floppy or flattened. Right? So an artery is very easily to distinguish because it has a nice round opening, whereas veins have more kind of a flattened opening, more kind of squished. And also you can see the tunica media layer of the veins is much thinner than the tunica media of the artery. So this dark purple line here is all smooth muscle in an artery. We just have a little tiny thin layer in the vein. Okay? But it does have a thicker tunica externa, right? So it's going to have those collagen fibers and elastic fibers to help support the structure overall of the vein. So these large lumens and thin walls are going to make veins very good storage vessels. So sometimes veins are referred to as capacitance vessels or blood reservoirs because they're capable of holding up to roughly 65% of our blood supply. So this is just showing relative proportions of blood volume throughout the entire cardiovascular system. So at any one time, the heart has roughly 8% of the blood volume, capillaries about 5% at the tissues, pulmonary blood vessels 12%, systemic arteries and arterioles 15%. Now you can see the veins and venules are going to contain more than half, right? So about 60% of our total blood volume at one time. So veins have lower blood pressure than the arteries because they don't have the force of the heart contraction behind them propelling the blood. So once the blood's made it into the venous system, it's kind of lost its momentum coming off the heart. So they have to have a few adaptations to make sure that the blood fully returns all the way to the heart. So one adaptation, of course, would be those large diameter lumens, so those large open lumens, to offer as little resistance as possible. So we don't want any hindrance to the blood flow on its way back to the heart. A few other adaptations we see with veins are venous valves. So these are only present in veins. So they're present to prevent backflow of blood through the blood vessel. So similar to the valves of the heart to ensure that one-way direction of flow, the valves of the blood vessels ensure the blood's only going to flow in one direction. Venous sinuses are another adaptation. So these are more flattened like veins with extremely thin walls composed only of an endothelial layer. So one example would be the coronary sinus of the heart and the brain, the dural sinuses of the brain here. So this is showing all these kind of flattened veins around the surface of the brain that's going to collect deoxygenated blood and return it to the heart through the jugular vein down to the superior vena cava. Okay, so just another comparison, summary of arteries and veins. So again, the elastic tissues are much higher in the arteries than in the veins, as well as smooth muscle. They have roughly the same amount of endothelium, and the veins, larger veins, are going to have more actual fibrous connective tissues on the outside. So looking at diameters, right, the different types of vessels. So the elastic arteries are going to have the largest diameters of the artery, so things like the aorta coming right off of the heart, okay? Muscular arteries, and then the arterioles. So with the capillaries and the veins, the capillaries are by far the smallest of all the blood vessels, right? So some of them are so microscopic, blood cells have to flow through single file. Right? So then from there we get to the venules, about 20 micrometers in diameter, up to the larger veins, which can be up to 5 millimeters in diameter. So homeostatic imbalance you can have with blood vessels and veins, pretty common, is varicose vein. So this is where the vein becomes dilated due to an incompetent or a leaky valve. So blood kind of backflows in the vein and the valve and starts to kind of pool, which causes the vein wall to become distended and enlarged. So some factors that can contribute to varicose vein formation include things like just heredity, genetics, or any condition that would hinder venous return. So standing for long periods of time, obesity, pregnancy, anything that makes blood kind of pool in the lower limbs is over time going to weaken those valves. Okay, so now shifting gears into some physiology, things like blood pressure. So just start with a few definitions of terms. So blood flow is the volume of blood that flows through the vessel or an organ or entire circulation over a given period. So we measure blood flow in milliliters per minute. So you can think of it kind of like cardiac output, but for the entire vascular system. Okay? So overall, it's relatively constant at rest, but it can vary based on individual organ-based needs. So say if you're exercising, right, so that would change the blood flow to your skeletal muscles and your heart and your lungs. So blood pressure is a measure of the force exerted on the wall of a blood vessel by the blood. So we measure this in millimeters of mercury. So we measure the systemic blood pressure for the larger arteries near the heart. And that blood pressure gradient as it goes throughout circulation is what's going to help keep the blood flowing from those higher to lower pressure areas, as well as propelling the blood into the tissues. So when we look at resistance, or sometimes called peripheral resistance, this would be anything that is opposing the blood flow. Okay, so it's going to resist blood flow. So basically it's a measure of the friction the blood's going to encounter with either the blood vessel walls or just generally in the systemic circulation. So three important factors for resistance include blood viscosity, or the thickness of the blood, total blood vessel length. So essentially the longer the blood has to travel, it's going to lose some of its momentum and pressure. And then blood vessel diameter. So we mentioned earlier with the highway analogy. So the more lanes of traffic you have open, right, the faster and the smoother things will flow. And vice versa, if we close lanes of traffic, we decrease the diameter or the size of the vessel, that's going to slow down the flow and increase that resistance. All right, so again, blood viscosity, just the thickness or stickiness of the blood due to things like the formed elements, blood cells, and plasma proteins. So the greater the viscosity, the less easily molecules are able to slide past one another. So that's going to increase the resistance and slow down the blood flow. So the blood gets kind of sludgy and doesn't flow as smoothly or as quickly through the blood vessels. So again, blood vessel length, the longer the vessel, the greater the resistance encountered, just because while it's traveling through the blood vessel, it's going to kind of lose its momentum. It's also going to drag some against the wall of the vessel, which slows it down. So ultimately, once it gets to the end of the vessel, its pressure and velocity is much slower than when it first left the heart. Blood vessel diameter probably has the greatest influence on our resistance to flow. So blood vessel diameter changes frequently, whereas viscosity and blood vessel length are relatively constant, or they don't change as rapidly. So blood vessel diameter can be altered within fractions of a second, right? Whereas viscosity and blood vessel length would take a little longer to take effect. So as the blood flows through the blood vessel, the fluid closer to the walls is going to move a little bit slower because it's going to have a little bit of drag against those blood vessel walls than the blood in the middle of the lumen or the tube. So this is called laminar flow. So you can see just a little bit slower on the edges and faster in the middle. So the resistance to blood flow is going to vary inversely or oppositely with vessel radius. So if the radius or the size of the vessel increases, right, so we open up another lane of traffic, so that's going to decrease resistance and vice versa. Right, so this is showing, so we have our normal blood flow, and then we encounter a decrease in our vessel diameter, which is going to kind of slow down and bottleneck the blood flow, where then it could become more turbulent. So the small diameter arterioles are more important determinants for this resistance factor. Since they're smaller, they're able to change their radius more drastically, so they could potentially completely close off those blood vessels, in contrast to the larger arteries that don't really change that often. So they're not able to constrict enough to close off the vessel or really change the resistance a whole lot. Any type of abrupt change in the vessel diameter or obstacles like atherosclerosis in the blood vessels can also increase resistance. So it can interrupt our normal laminar flow and cause more turbulent, irregular flow, which is also going to increase resistance. So say this is a fatty plaque in an artery. So we have our normal smooth laminar flow, which is going to encounter this plaque, and then it's going to kind of flow over it and around it. So now the blood is all more turbulent and not flowing as smoothly or as straight. So the relationship between blood flow, blood pressure, and peripheral resistance is shown by the equation flow is equal to pressure gradient divided by resistance. So you don't need to know the equation. Really, I'm not going to make you do any math, but just showing that it's an inverse relationship to resistance and a proportional relationship to blood pressure. So the higher the blood pressure, right, so the flow is also going to increase. Whereas if we increase the resistance, that's going to decrease our blood flow. So ultimately, resistance is going to be more important for influencing our blood flow because it's more easily changed by just altering those blood vessel diameters. Okay, so this is trying to show the relationship between blood pressure and velocity. So at the aorta, when it first leaves the heart, we have that full force of contraction behind that blood leaving the heart in the aorta. So we have the highest pressures and the fastest velocities. Right? So you see it's kind of a proportional relationship. So as blood pressure decreases, the velocity or the speed of the blood flow is going to decrease roughly the same amount. So when we look at systemic blood pressure, this is being generated by the pumping of the heart to generate that blood flow. Right? So again, pressure results when the flow is opposed by resistance. So the systemic pressure is going to be highest in the aorta, and then as we go throughout the circulation, it's going to steadily decline. So when we look at arterial blood pressure, we have to look at the systolic pressure. So your systolic pressure is the pressure that's exerted in the aorta during ventricular contraction. So remember last time when we talked about systole and diastole, so contraction and relaxation. So this is just measuring the pressure generated by those contractions and relaxation. So systolic pressure is the pressure generated during ventricular systole. So the left ventricle is going to pump that blood into the aorta and cause it to stretch. Remember, it's an elastic artery, so it has a lot of elastic fibers, so it's capable of that stretch and recoil. So average numbers for systolic pressures in adults is about 120 millimeters of mercury. Okay. So then the diastolic pressure would be the pressure during heart relaxation, right, or diastole. So this would be the lowest level of pressure. So this would be your bottom number for blood pressure reading. Okay? So that's why it makes sense that your diastolic should always be a lower number than the systolic. Pulse pressure would be the differences between your systolic and diastolic. So that can be felt as the pulse, so the throbbing of the arteries due to the difference in those pulse pressures. So you can feel the arteries expanding and recoiling during systole and diastole. Okay. So the mean arterial pressure is kind of the average between the systolic and diastolic. So it's the usable pressure that's going to actually propel and push the blood through into the tissue spaces. So when we want to monitor the efficiency of circulatory system, we look at things like vital signs. So pulse, blood pressure, respiratory rate, body temperature, everything that's vital for life, right? So if you don't have a pulse or blood pressure, then you're dead. So these are the most vital signs to look for when checking circulatory efficiency. So when we take a pulse, you're essentially, like I said, feeling the difference between those pulse pressures, so an expansion and recoil of the arteries. So these are just some common places where pulses can be detected. So the radial pulse, radial artery is the most commonly used. There are also some other ones, so the carotid artery is another common one, brachial artery, where you can feel the pulse under the skin. These pressure points are also areas where the arteries are close to the body surface. So in the event of hemorrhaging or injury, we can put compression on these arteries to kind of stop or hinder the blood flow to that injured area. So the process of measuring blood pressure is called the auscultatory method. Right? So you've all had this done before when you go to the doctor's office. Okay? So essentially you put the blood pressure cuff on the arm, okay, above the elbow. So as we inflate the cuff, we're going to inflate it so tight that it completely closes off and stops the arterial blood flow. Okay. So at this point, no sound can be heard through the stethoscope, so we don't hear any blood flow or anything through that artery. Okay. So then we'll slowly release the pressure while we're listening. So as soon as the blood starts to flow again, you'll hear the first sounds. Right? So these are sometimes called the sounds of court cough. Okay. So whatever pressure the blood pressure cuff is at when you hear that first sound of blood flowing again through the artery, it's going to be your systolic pressure. So like I said, it's usually 120. Right? So then you'll continue to slowly release the pressure of the cuff until you no longer hear any blood flow. So the first sounds we heard were the turbulent blood flow through the artery. So once the artery is completely open again, the blood flow should return to normal and have no sound. So the pressure at which the sound disappears, so when blood flow becomes silent again, is your diastolic pressure. So looking at capillary blood pressure, these have some of the lowest blood pressures in the circulatory system, ranging from 35 millimeters mercury to 17 millimeters mercury. So we want to have a very low blood pressure in the capillaries because a high pressure could potentially rupture these thin, fragile capillary vessels. And being that the capillaries are permeable and they're exchanging nutrients and gases and things with the tissues, we don't want the blood to pass through too rapidly. We want it to take its time passing through so that we get maximum gas and nutrient exchange. So venous blood pressure is not going to change a whole lot from during the cardiac cycle. It has the lowest pressure gradient, only about 15 millimeters of mercury. So this is why if you just get a cut, you cut a vein, it's kind of just a slow leaking or oozing of blood. Whereas if an artery is cut, the blood is going to spurt out because the pressure is much higher in those arterial vessels. So the low pressure in the venous system is due to several effects of peripheral resistance. So the body's going to require some adaptations to assist with venous return despite this low pressure. So we don't have enough pressure on our own. The blood pressure itself is not enough to return the blood to the heart. So remember, especially coming from the inferior vena cava, right, the lower portion of the body, we have to work against gravity as well. So some factors that aid in venous return include a muscular pump. So as our skeletal muscles contract, they're going to squeeze the blood vessels and kind of milk the blood back toward the heart. So they squeeze the blood a little bit at a time, and then the valves we mentioned earlier are going to prevent any backflow. So we squeeze and get past this valve, and we squeeze again to get past this valve, right, all the way until we make it back to the heart. The respiratory pump is going to work the same way as the muscle pump, but with during breathing, the squeezing of the abdominal veins as the chest cavity and the lungs expand and recoil. It's going to also help to squeeze the blood back toward the heart. And then the third factor would be sympathetic venoconstriction. So remember, the sympathetic nervous system is your fight or flight. So it's sympathetic impulses will cause vasoconstriction. It causes the smooth muscle in that tunica media to contract, which would increase blood pressure. So regulation of blood pressure, when we have to maintain blood pressure within those homeostatic ranges, it's going to require cooperation of the heart, the blood vessels, as well as the kidneys. And being the control center of the body, all of these processes are supervised and kind of monitored by the brain and the central nervous system. So we have three main factors to help regulate our blood pressure. So we can look at cardiac output, peripheral resistance, and blood volume. So blood pressure is going to vary directly or proportionally with cardiac output and peripheral resistance and blood volume. So remember from last chapter, cardiac output equals stroke volume times heart rate. So if our mean arterial pressure is equivalent to cardiac output times resistance, right? So the actual blood pressure that is usable blood pressure, right, that's used to propel blood into the tissues is a factor of how much blood the heart is putting out times the resistance that blood flow is going to encounter. Right? So essentially it's the same thing as saying the stroke volume times the heart rate times the resistance. Right? So again, I'm not going to make you do any math or anything with these equations, just trying to show the relationship between these different factors. So essentially anything that increases your stroke volume, heart rate, or resistance is also going to increase your mean arterial pressure. So we talked about last chapter, the stroke volume can be affected by venous return, so that end diastolic volume. So the more blood we can fill into the heart chambers, right, the more blood we can pump out into circulation. The heart rate can be maintained by medullary or higher brain centers. So something like the sympathetic fight or flight system can influence heart rate. Resistance, we said, was affected mostly by vessel diameter. So we can constrict or dilate that vessel, right, to increase or decrease the flow and the pressure. Okay. So in mean arterial pressure, it's a factor of cardiac output and peripheral resistance. Right? And we talked about last chapter, cardiac output is just stroke volume and heart rate. So how much blood is pumped out of each ventricle, right, per each beat, and then how many times the heart beats per minute. Okay. Peripheral resistance is a factor of the diameter of the blood vessels. Right? So again, the more open the blood vessel, the less resistance. So it'll increase the flow. So if we decrease the diameter of the blood vessels, that's going to increase our resistance. Increase in blood viscosity or thickness, right, can increase resistance. So the blood flow is a little more sluggish. Or blood vessel length. So the longer the blood has to travel, it kind of loses momentum and pressure the longer it travels. So the longer the blood vessel length, the increase in resistance the blood flow will encounter. So we can regulate blood pressure by a few different methods. So We can have short-term regulatory methods, either using neural controls in the brain and the nervous system or hormonal controls. We can regulate and influence blood pressure more long-term solutions using renal or kidney controls. So there are two main neural mechanisms for short-term regulation of peripheral resistance. So these include things like maintaining mean arterial pressure by altering blood vessel diameter, which thereby alters resistance. So this example says if blood volume drops, so then all vessels would constrict except those to the heart and brain. So we'll prioritize the most vital organs first, so they get first priority on blood flow. We can also alter blood distribution to different organs in response to different demands. So like during exercise, your skeletal muscles, your heart, your lungs are all going to be demanding higher blood distributions. So these neural controls operate via a reflex arc. So it's going to involve some things like cardiovascular center of the medulla. So remember, the medulla was kind of the primitive brain that's primarily concerned with just keeping you alive. So things like heart rate, breathing rate, things like that. Baroreceptors are pressure receptors located within the blood vessels. Chemoreceptors monitor blood chemical composition, pH, ion concentrations, things like that. And then higher brain centers, so something like maybe your fight or flight system. So if something scares you, right, then your heart rate, your blood flow may increase. So the cardiovascular center of the medulla is composed of those sympathetic nerve fibers, right? So that fight or flight nerve fibers, consisting of cardiac center. So this is either inhibitory or acceleratory center. So we can either speed up or slow down the heart rate, which would then influence our blood flow and blood pressure. The vasomotor center is kind of a steady, constant impulse of those sympathetic fibers to the blood vessels. So this just means that all blood vessels have a slight vasomotor tone, or they're always slightly constricted. Okay, so this is also going to receive input from those baroreceptors and chemoreceptors and higher brain centers. So they're all kind of interconnected. Baroreceptor reflexes, so those pressure receptors located in the carotid arteries, the aorta, and some larger arteries of the neck and thorax. So if blood pressure gets too high, that's going to stimulate those baroreceptors to increase input to that vasomotor center. So we can essentially inhibit those cardioacceleratory centers and stimulate the inhibitory center, so resulting in a decreased blood pressure. So essentially, we're going to decrease the heart rate and potentially undergo vasodilation. Okay, because we want to lower that blood pressure. So if blood pressure gets too high, it can damage the blood vessels in the heart. Okay. So vasodilation is caused from that decreased output from our vasomotor center. So instead of telling the blood vessels to always stay slightly contracted, we turn off those signals so now they can relax and dilate a little bit more. Okay. So vasodilation is going to reduce that resistance, which is going to reduce the blood pressure. Okay. When we have a venodilation, we can causes a decrease in the venous return and therefore the cardiac output. So if we have less blood returning to the heart, that means we have less blood that the heart can pump into circulation. Right. So again, decreased cardiac output would be some of those inhibitory signals from the sympathetic system, right? Or stimulation of the parasympathetic. Right. So remember, sympathetic and parasympathetic are opposite. So whereas sympathetic is your fight or flight, that would increase heart rate and blood pressure, parasympathetic is the rest and digest. So it reduces heart rate and contractility, or that force of contraction, which thereby decreases cardiac output and blood pressure. Okay, so this figure is just showing how these baroreceptors can work in either cases of high blood pressure or low blood pressure. So if our stimulus is an increase in blood pressure above the normal homeostatic range, those baroreceptors will be stimulated. They'll detect that pressure and notify our control center in the brain. Right? So those impulses from the baroreceptors are going to stimulate the inhibitory center of the cardiovascular part of the brain. So ultimately, we'll turn down those sympathetic impulses. Right? So a decrease in sympathetic activity would decrease heart rate, contractility, and cardiac output. So we're slowing everything down. Right? We're also going to reduce the rate of that vasomotor impulse. So we're letting the vessels relax a little bit and dilate to decrease resistance. Right? So blood flow would flow more freely. So finally, a decrease in the cardiac output and resistance is going to return the blood pressure back to the normal level. Okay. So opposite end of the spectrum, if we have a decrease in blood pressure, so it falls below normal range, again, baroreceptors will detect that, notify the cardiac centers of the brain. Okay. So that's going to activate the acceleratory center and stimulate our vasomotor fibers. Right? So an increase in sympathetic activity is going to increase heart rate, increase contractility, and thereby cardiac output. Okay? We'll also stimulate those sympathetic fibers in the smooth muscle of the tunica media for vasoconstriction. So we close off the blood vessel more, so we decrease the diameter and increase the resistance. Right? So we're going to slow down the blood flow to increase its resistance and pressure. So finally, an increase in our cardiac output plus resistance returns blood pressure back to the normal homeostatic level. Okay. Chemoreceptors are going to detect, like I said, chemical composition of the blood, so things like oxygen, carbon dioxide levels, or pH. Okay. So this could be caused or cause increased blood pressure by either stimulating the cardiac center of the brain to increase cardiac output, or stimulating the vasomotor center to increase that vasoconstriction. Right? So if we increase the amount of blood being pumped by the heart and we increase the resistance of the blood flow, that results in an increase in blood pressure. So those higher brain centers are also going to operate under reflex arcs to help regulate blood pressure, and these are found in the medulla. Again, so kind of the primitive part of the brain just with keeping us alive, our major vital body functions. Okay. So the hypothalamus and the cerebral cortex, so kind of your higher brain centers, can modify arterial pressure by sending information to the medulla. Right? So example, during stress, right, you'll have more sympathetic impulses, right, and increased heart rate. So we can also redistribute blood flow during exercise or stressful situations to the vital organs. So hormones can also regulate blood pressure in the short term via changes in either peripheral resistance or potentially long-term regulation by changing the blood volume. So generally hormones are considered short-term mechanisms because they're only going to have an effect as long as the hormone's being produced. But remember from chapter 15 that most hormones operate under a negative feedback. So once the hormone does its job to restore blood pressure, then generally we'll stop producing that hormone. So the effect is only going to last as long as the hormone's being produced. So some hormones that can influence blood pressure that we've already talked about, just to review. So the adrenal medulla hormones, epinephrine, norepinephrine, those catecholamines, right, from the adrenal gland. So these are like your adrenaline, your fight or flight hormones that are going to increase cardiac output as well as vasoconstriction. Angiotensin II stimulates vasoconstriction, so that was part of the renin-angiotensin-aldosterone mechanism, so kind of that intermediary. So it stimulates vasoconstriction to kind of temporarily raise blood pressure while we wait for the aldosterone to be released and work. ADH, antidiuretic hormone, can cause vasoconstriction. Atrial natriuretic peptide, we talked about, actually decreases blood pressure. So it's antagonistic to the aldosterone, right? So aldosterone was released in instances of low blood pressure. Atrial natriuretic peptide is released in high blood pressure. Okay, so cells in the atrial wall of the heart will detect that increase in pressure and release this ANP hormone, which ultimately causes decreased blood volume by secreting more sodium and water. So again, just to review some effects of these hormones. So epinephrine, norepinephrine, angiotensin, ADH, and aldosterone all have increasing effects on blood pressure, right? So the atrial natriuretic peptide is the only one that works to lower blood pressure. Right? So they do this by either altering cardiac output, so things like heart rate. We can alter peripheral resistance with vasoconstriction or vasodilation. Or we can alter blood volume, so either by increasing or decreasing our water loss through urine. Right? So for these, it would target the kidney cells. Right? So long-term mechanisms, looking at renal regulations or kidney regulation mechanisms. So our baroreceptors, those pressure receptors, are able to quickly kind of adapt to blood pressure changes, but like I said, they're only effective in the short term. So they're not very effective for long-term regulation. So to have a more long-term effect on blood pressure, we need to alter blood volume. So things like vasoconstriction or heart rate are more short-term effects, so we can't maintain that for prolonged periods of time, right? So with blood volume, it takes a little bit longer to alter because if we want to decrease or increase blood volume, we have to rely on the kidneys, which control our water balance, right, by filtering the blood and removing water and solutes. So kidneys can regulate our arterial blood pressure by one of two mechanisms. So they can either do this directly through the kidney, so just filtration and secretion, urine formation, or indirectly. So this would go back to that renin-angiotensin-aldosterone mechanism. So the direct renal mechanism's most straightforward, right, and direct. So it's going to alter blood volume independently of hormones. So it's self-contained within the kidneys all by themselves. So an increase in blood pressure or blood volume would cause more urine formation. So we're going to lose blood volume and reduce blood pressure. Vice versa, if we have a decrease in blood pressure or blood volume, the kidneys are going to try to conserve more of that water by decreasing urine output. So ultimately that water is going to be returned to the blood, which increases blood volume and raises blood pressure. So the indirect mechanism, so not as straightforward, but it is very efficient, this renin-angiotensin-aldosterone mechanism. So a decrease in blood pressure is going to stimulate release of renin from the kidneys. So renin is going to catalyze some enzymes to form, leads to the formation of angiotensin II. Okay? So angiotensin is especially efficient at increasing blood pressure by a few different mechanisms. So 4 ways that angiotensin II helps to stabilize blood pressure and extracellular fluid. So first of all, it can stimulate aldosterone release, right? So it can stimulate the adrenal cortex to secrete aldosterone. It can also increase secretion of ADH, so we conserve more water. So the aldosterone and the ADH together lead to an increase in water reabsorption by the kidneys, which ultimately results in an increase in our blood volume and blood pressure. Angiotensin II can also stimulate the thirst center, increase your sense of thirst in the brain. So we increase our water intake, which increases blood volume and pressure. And just by itself can act as a vasoconstrictor. So it increases our peripheral resistance, which increases blood pressure. So kind of a four-way attack of increasing blood pressure through this mechanism. So essentially the goal of blood pressure regulation is just to keep the blood pressure high enough so the blood and fluids are able to perfuse or pass through to the tissues, but we don't want it so high that the blood vessels will become damaged. So again, just kind of keeping everything within that narrow homeostatic range. So some examples of blood pressure to the brain is too low. So the blood or the brain cells aren't receiving enough blood and oxygen. The person can lose consciousness, right? Or vice versa, if the blood pressure in the brain is too high and it damages those blood vessels or ruptures them, that would lead the person to have a stroke. Okay, so just a summary now of all of the factors that can influence our blood pressure. So primarily cardiac output and peripheral resistance, right, are our main two. So there's several ways that we can alter our cardiac outputs and peripheral resistance. So increased activity of the muscular and respiratory pumps is going to increase the amount of blood returned to the heart, which means we have more blood we can pump out for our stroke volume, which increases our cardiac output. So if we decrease atrial natriuretic peptide, right, so remember this was the one that decreases blood pressure. So if we want to increase blood pressure, then we'll turn off the ANP. So that's going to increase conservation of sodium and water by the kidneys, which leads to an increase in blood volume, right? Increased venous return, stroke volume, and cardiac output. So if we have fluid loss from hemorrhage or excessive sweating, that's going to decrease our blood volume and blood pressure, which would be detected by the baroreceptors. Okay? So these baroreceptors can stimulate the acceleratory cardiac centers in the brain, right, which can increase our heart rate, right, and cardiac output. Exercise, stress, trauma can affect the chemical composition of the blood, right, which would be detected by chemoreceptors, and again activate those cardiac centers in the brainstem to increase heart rate, cardiac output. So we can also alter the diameter of the blood vessels. So things like epinephrine, angiotensin II, ADH, increasing that vasomotor tone, right? So ultimately a decrease in diameter of blood vessels, right? So we're closing a lane of traffic. It's going to increase the resistance and slow down the flow. Okay? So dehydration or high hematocrit, so a high concentration of red blood cells, so a thicker blood, increases the viscosity, which can also increase resistance, and just increase in body size. So we have to have longer blood vessels to reach all of those tissues, which also contributes to an increase in peripheral resistance. So some homeostatic imbalances with blood pressure, things like hypertension, right? So this would be where the blood pressure is abnormally high. Right? So anything over, say, 140/90 would be considered hypertension. Okay? So prolonged hypertension can cause things like heart failure, heart attack, vascular disease, renal failure, or even stroke. Because essentially the heart's going to have to work harder to overcome that pressure and that resistance. Right? Or think the afterload when we talked about last chapter. Right? So the heart has to work harder and pump harder to overcome that resistance. So as a result, the myocardium, the heart muscle, can enlarge or become weakened and flabby. Okay, so this process can also accelerate atherosclerosis just by kind of irritating and adding increased pressure to those vessel walls. So the opposite end of the spectrum would be hypotension. So this would be a low blood pressure, generally around 90/60. So usually it's not as much of a concern as long as we still have adequate blood flow to the tissues. Highly trained athletes can generally have lower blood pressure, so it's often associated with lack of cardiovascular illnesses and longer, healthier lives. So one type of hypotension is called orthostatic hypotension. So this would just be when you stand up too fast and you get a little dizzy and lightheaded. So it's just a temporary low blood pressure when suddenly rising from a sitting or reclining position. So kind of gravity slows down the flow of blood back up to the brain for just a few seconds. Chronic hypotension can be a hint at poor nutrition or a sign for other underlying diseases like Addison's disease or hypothyroidism. And acute hypotension would be an important sign of circulatory shock. So circulatory shock occurs when blood vessels aren't able to be adequately filled or circulate blood normally. So ultimately, we are not able to meet the needs of the tissues, and the tissues can start to die. So there's three types of circulatory shock. A hypovolemic shock would result from just a large scale blood loss. So hemorrhages, diuresis, plasma loss through burns, diarrhea, vomiting, decreased body fluids. A vascular shock would result from extreme vasodilation or decreased peripheral resistance. So blood pressure just gets so low, resistance is so low that the pressure isn't enough to leave the capillaries and enter into the tissues. Right? So remember, we have to have that average mean arterial pressure to kind of push the fluid and the blood and the oxygen and nutrients from the capillaries into the tissues. Cardiogenic shock results when the heart's inefficient and can't sustain adequate circulation. So the heart's not able to pump enough blood or with enough force to supply circulation. So when looking at blood pressure, we have to look at tissue perfusion. Right? So tissue perfusion is just the blood flow through to the body tissues. So delivering oxygen and nutrients, removing waste from the tissue cells, also gas exchange in the lungs, absorption of nutrients in the digestive tract, and urine formation by filtration in the kidneys. So generally the rate of blood flow is just the right amount to provide proper function to that tissue or organ. Right? So remember, the capillaries have those precapillary sphincters that they're able to kind of control how much blood and when is going to enter specific tissue spaces. Right? So looking at control of blood flow, so the rate of blood flow is going to be controlled by extrinsic and intrinsic factors. So again, extrinsic would be something that's outside the blood vessel itself. So looking at the sympathetic nervous system or hormones that influence blood flow through the body by acting on the arterial smooth muscle for either vasoconstriction or vasodilation. So neural or hormonal helps to maintain mean arterial pressure, redistributes blood during exercise. Right? So an increase in the sympathetic tone would cause vasoconstriction. Right? So your fight or flight, we're going to increase blood pressure. A decrease in the sympathetic impulse would cause vasodilation. So we're going to let the smooth muscle relax and open up those blood vessels. Okay? So the hormonal effects. So again, remember, atrial natriuretic peptide, the ANP, were the only hormones that have a decreasing effect on blood pressure by causing vasodilation. All the other hormones we talked about, so angiotensin, ADH, epinephrine, are going to cause vasoconstriction to increase blood pressure. So Intrinsic controls or autoregulatory mechanisms are ways that the blood vessels can adjust themselves on their own to meet specific tissue requirements. So like the arterioles that feed into the capillary beds are capable of modifying their own diameters or altering those precapillary sphincters to let blood in or out of the capillary bed. Organs can also regulate some of their own blood supply by varying their resistance of their arterioles. So, and if you're exercising your skeletal muscle, arteries, arterioles will dilate to increase the blood flow to those tissues. So, example, redistribution of blood during exercise. So during rest, these skeletal muscles only receive about 20% of total blood in the body, but during exercise, they can go up to 70%. Right? So skeletal muscles have very high metabolic demands and require lots of blood and oxygen and nutrients. So again, intrinsic controls would be the skeletal muscle arterioles dilating to increase the blood flow. Extrinsic controls could be decreasing blood flow to other organs, such as the kidney or the digestive organs. So we can prioritize blood going to the skeletal muscles. Right? So at rest, we're sending 1,100 and 1,400 mils per minute to the kidneys and the abdomen, the digestive organs. But during exercise, the skeletal muscles kind of take priority over those. Right? So especially if you are in a fight or flight situation, right? So your sympathetic nervous system is activated. So if you're running away from an axe murderer, right? So digesting your lunch or going to the bathroom can wait, right? So we're going to focus all of our excess blood and energy to these muscles so we can fight or run away. Right? So during exercise, kidney blood flow goes down to 600, right, as well as in the abdomen. So again, these autoregulatory mechanisms are intrinsic mechanisms to help regulate blood flow to a specific area. So sometimes it's called reactive hyperemia. So we're going to increase the blood flow to an area in reaction to these intrinsic factors. So there's two types of intrinsic mechanisms that can contribute to this auto-regulation of blood flow, and those are metabolic controls or myogenic controls. So metabolic controls are going to increase the metabolic activities, or an increase in tissue metabolic activities is going to result in things like lowered oxygen levels, increase in waste products like carbon dioxide, hydrogen, potassium. So there are also some local chemicals produced that can cause direct relaxation of either the arterioles or those precapillary sphincters. So things like nitric oxide is a powerful vasodilator that's released by some of those endothelial cells within the blood vessel. Okay, so again, so if the tissue or the blood has low oxygen, we can dilate the blood vessel to increase the blood flow to those tissues and hopefully increase the oxygen levels. Myogenic controls, remember myo means muscle, so this would be local response to the vascular smooth muscle in response to changes in the arterial pressure and blood pressure, because we want to keep tissue perfusion relatively constant so the tissue stays well supplied with oxygen and nutrients, and also we don't damage any of the tissue cells. So this would be a factor or response to muscle stretch of the vessel wall. So an increase in stretch, right, smooth muscle stretch would respond by kind of constricting and decreasing the blood flow to that tissue. Okay? So a reduced stretch would mean a decrease in blood pressure and cause less stretching as normal. So then the blood vessels would dilate, right, to increase blood flow to the tissue. So finally, long-term autoregulatory mechanisms occur generally when the short-term mechanisms aren't able to meet tissue requirements. So because it's long-term, it does take a little bit longer to take effect. So it may take weeks or even months to finally increase the blood supply. So generally, this is going to be done through the process of angiogenesis. So existing vessels enlarge, or we increase the number of blood vessels going to a region. So this can be commonly seen in heart, coronary vessels when one is blocked. So the heart tries to compensate for that by making new blood vessels or enlarging the already existing blood vessels. Same thing with people in high altitude areas, so where the oxygen content of the atmosphere is pretty low. The body tries to compensate for that by making more blood vessels or larger blood vessels, so we can deliver more blood to the tissue spaces. Alright, so finally looking at just some blood flow to special areas. So skeletal muscles, blood flow is going to vary with the skeletal muscle activity. Right? So like I said, at rest, it's going to have a relatively average flow of about 1 liter per minute. So during activity or exercise hyperemia, the blood flow is going to increase to those skeletal muscle fibers in proportion to the metabolic activity. So as the skeletal muscles are working, they're going to be using a lot of oxygen and making a lot of carbon dioxide. So we need to bring them more oxygen and remove that carbon dioxide and those other metabolic wastes so they don't build up in the tissues. So exercise in skeletal muscle is going to decrease oxygen, increase carbon dioxide, other waste products. Leads to vasodilation of those arterioles. So we bring more blood to the tissue to increase the blood flow, resulting in active hyperemia. Okay. So blood flow to the brain is relatively constant because brain cells do not tolerate lack of oxygen or ischemia. So flow averages about 750 mils per minute. As you see, even during exercise, it's going to remain pretty much constant no matter what. The brain's very vulnerable under extreme blood pressure changes. So we want to keep it as constant as possible. So if blood pressure falls below, or mean arterial pressure falls below 60 millimeters of mercury, can cause what's called syncope or fainting. Blood pressures above 160 can result in cerebral edema or even potentially stroke. So the skin is also a special area where blood can flow. So functions of blood flow through the skin include supplying nutrients to the cells, regulating body temperature, and also providing a blood reservoir. So see, the skin is capable of holding several times more, about 4 times as much blood during exercise as during rest. Right, so blood flow in the lungs is a little bit different. So remember, the pulmonary circuit we said was kind of opposite from the systemic circuit. So first of all, the pathway is very short. So we're just going from the heart to the lungs and back. So in this case, the arteries and the arterioles are structurally more like the veins and the venules in that they have thinner walls and they have larger lumens. Okay? Also, they're carrying deoxygenated blood. So remember we said arteries carry blood away from the heart, veins carry blood toward the heart. So the only exception would be the pulmonary system. So deoxygenated blood leaves the heart and goes to the lungs to pick up oxygen, and then it returns to the lungs with the pulmonary veins. That is oxygenated. So generally veins have deoxygenated blood except the pulmonary circuit. Also, the resistance and pressure are much lower than the systemic, again, because it doesn't have to travel very far, so we don't need a whole lot of pressure behind it leaving the heart. Also, so the autoregulatory mechanisms are also opposite. So low oxygen levels would cause a vasoconstriction. So that would allow blood to spend more time in the lungs and give it more time to pick up oxygen, right? Whereas, say, in skeletal muscle, if we have low oxygen levels, it would cause a vasodilation, right? So that we bring more blood and oxygen to the tissue. Right? But everything in the pulmonary circuit is going to be the opposite or backwards. Okay? So, vice versa, high levels of oxygen would promote vasodilation. Right, so if the blood already has a lot of oxygen, it doesn't need to spend a whole lot of time in the lungs. We'll dilate the vessel so it can flow back to the heart more quickly. So blood flow through the heart is going to be influenced by the aortic pressures and ventricular pumping. So during ventricular contraction, those coronary arteries are going to be compressed. So then during relaxation, diastole, the blood isn't going to be forced through those coronary circulations. It can also dilate in response to exercise and vasodilators. So blood flow to the heart may increase 3 to 4 times during exercise. Right? So we don't want our heart muscles to get fatigued, right, or stop contracting, because then we would die. So we need a steady, constant blood supply through that coronary circulation. So looking at capillary exchange and the velocity of blood flow. So as we go through the systemic circulation, we see the velocity of the blood flow is going to change. So it's going to be fastest in the aorta, right? So again, when it first leaves the heart, it's got the most force and pressure behind it. And then it slows down in the capillaries, right? So again, we don't want high pressure in the capillaries because we want to give the blood enough time to exchange gases and nutrients with the tissues, and we don't want to rupture or damage those small, fragile capillary vessels. So then it picks up a little bit more once we get into the venous system, but still never reaching velocities like in the arterial system. So this is showing that the velocity or speed of blood flow is going to be inversely related to the cross-section of the vessel area. So capillaries are going to have the largest section, cross-section area or surface area, right? So instead of having just one vessel, now we have all these branching, kind of interweaving networks of vessels. So it's going to cover more surface area. So that also means it's going to slow down the flow. Right? So instead of all the blood flowing in one direction, now it's being split off in all these different directions and forks. So vasomotion, we look at capillary blood flow. It's just kind of that intermittent flow through the capillary. So the precapillary sphincters will kind of open and close kind of rhythmically or intermittently to allow blood to seep in just a little bit at a time. So we don't have the full force rush of blood entering into the capillary beds. Okay. So capillary exchange of gases and nutrients. Many molecules are going to pass by just simple diffusion between the blood itself and the interstitial fluid. So remember from Anatomy 1, diffusion is just moving down the concentration gradient. So we're going from high to low concentration. So say the tissue spaces have a low concentration of oxygen and a high concentration of carbon dioxide, whereas the blood inside the capillary has high oxygen levels and low carbon dioxide. So the oxygen is going to diffuse out of the blood and into the tissues. The carbon dioxide will diffuse from a high concentration in the tissues to low concentration inside the blood, where it'll be carried to the heart and then the lungs to be exhaled. Okay. Another way that substances can pass through the capillaries are through these intercellular clefts. So water-soluble substances are able to kind of squeeze through these intercellular spaces to enter the tissues or the capillaries. Substances can also pass through these fenestrations or these pores and windows through the capillaries. And then finally, active transport. So this would be using vesicles. So things like proteins, larger molecules um, that can't pass through the clefts or via diffusion. So we package them in a little sac like a vesicle and then expel it into the tissue space.